recent works as example, and their preparation, characterization, and applications in biomedical field. Biomaterials, biomaterial-based nanoparticles such as dipeptides, proteins, and polysaccharides are increasingly used for nanomaterial synthesis because they are biodegradable, biocompatible, and non-toxic. These biomolecule-derived nanostructures enable precise drug delivery, molecular imaging, and targeted therapy, particularly in cancer and neurodegenerative disease. Dipeptide-based nanomaterials have some advantages of structural simplicity, such as well-defined chemical structure, predictability, and reproducibility. Fine-tuning and properties. And rational design approach. Among these biopolymers, proteins like albumin, lysozyme, and casein are particularly interesting due to their amphiphilic nature and structural flexibility, allowing controlling self-assembly into nanoparticles. I think it is worth emphasizing, according to my experience, proteins without quaternary structure are generally more suitable for nanomaterial preparations, nanoparticle preparations. Biomaterials exhibit remarkably self-assembly abilities, forming diverse nanostructures through non-covalent interactions like hydrogen binding, pi-pi stacking, hydrophobic interactions, etc. At the same time, change in environmental conditions such as pH, solvent composition also have effect on particle size, shape, and stability. The intrinsic biological functions of source proteins are often retained within the nanomaterial architecture, providing significant advantages in nanomedicine. For example, protein-based nanocarriers can transfer the drugs into cells via their receptors on the cell surface. If you use albumin having naturally binding sites to fatty acids, hormones, and drugs, it, it can interact with cells by EPR effects and active targeting through albumin receptors. And also, if transferrin nanoparticles are used, they are actively taken up by cells via transferrin receptors. And because of these properties, nanoparticles may sometimes not require additional surface modifications. As for preparation methods for protein peptide-based nanomaterials, we can generally talk about 5 main categories. First of this category is precipitation of natural proteins. Here, Protein molecules are precipitated by changing environmental conditions such as alcohol and pH, and stabilized with the help of crosslinkers after forming of nanoparticles. The second one, by formation of denatured proteins, when denaturation occurred, for example, by adding urea solution, denaturation process will begin, and denatured proteins aggregate via van der Waals forces requiring cross-linking for stability after forming nanoparticles. The third one, formation of protein nanoparticles in emulsions. In this technique, emulsion droplets formation creates protein nanoparticles by dispersing proteins in a hydrophobic solvent and evaporating the solvent after cross-linking. Self-assembly of protein polymer conjugates in another method. Here, in this type of nanocarrier system, nanoparticles having hydrophobic structure are covered with protein. And the last one, preservation of charged polymers and proteins. Here, nanoparticles are formed by creating a charge on proteins and mixing them with opposite charged polymers. And now let me give you information about characterization of nanoparticles. These characterization methods generally used for many types of nanoparticles, but some of these are specific for protein, especially for protein based nanoparticles, nanomaterials. For investigation of nanoparticles, surface and spectroscopic characterizations, size and morphological analysis and thermal property studies are done. The surface properties of nanoparticles determine biological interactions, drug release kinetics, and material stability, making comprehensive surface characterization critical for biomedical applications. Within this scope, surface charge, surface chemistry, surface area, and molecular structural analysis are carried out. Spectroscopic methods provide detailed information about chemical composition, functional groups, and molecular interactions in biopolymeric nanomaterials, essential for understanding their properties and behavior. In the scope of characterizations, FTIR and CD circular dichroism spectroscopy can reveal the secondary structure of protein, confirming the mildness of synthesized route. While dynamic light scattering and transmission electron microscopy confirming uniform spherical morphology, zeta potential analysis show the colloidal stability of particles having a stable surface charge. This type of characterizations generally used for all nanoparticles, especially circular dichroism, for example, used for protein-based nanomaterials. As for applications, these protein-based nanomaterials have shown promising results in several biomedical applications. Dipeptide and protein-based nanomaterials enable precise delivery of therapeutic agents to specific disease sites while minimizing of target effects, enhancing treatments efficiency and reducing systemic toxicity. The precision targeting capabilities of these nanomaterials significantly enhance therapeutic efficiency while reducing side effects, making them valuable tools for advancing therapeutic strategies for both cancer and neurological conditions. Biomaterial-based nanoparticles enable non-invasive monitoring through incorporation of imaging agents. They can be designed to accumulate at tumor sites, enabling enhanced contrast in imaging techniques like MRI or fluorescent imaging, thereby aiding in diagnosis and guiding treatment strategies. Now, let me talk about some studies on this subject in our group. Here you can see two types of nanomaterial preparations. In the first part, classic preparation of drug encapsulated protein-based nanoparticles, and second part, preparation of drug encapsulated protein-based nanomaterials by denatured protein. Classical nanoparticle formulations are formed by the entrapment of drug molecules. You can see here protein molecules and drug molecules. By entrapment of drug molecules between protein ones and finally obtained nanoparticles stabilized by cross-linkers. In denaturation or renaturation strategy, first step includes denaturation of proteins with some agents like URA, then conjugation of active agents with proteins. For example, you can use drug here, and you can conjugate these drugs with denatured proteins, and renaturation and formation of nanoparticles, including conjugated protein molecules, as you see here. In this method, you can encapsulate more active agent into nanoparticles when compared to classical ones. Or this study from my some studies here. Now we performed some experiments with encapsulation of drugs. For example, in literature, generally classical protein-based nanomaterials are referred, but and also the natural conditions with formation of nanoparticles is also have. But here you can see the classical formation of protein-based nanoparticles. There is a difference between these techniques. When you prepare the natural formation of protein for encapsulation of drugs, you can encapsulate more active agent, for example, drugs, and it is also for more releasing. This formulation is better when compared with the classical protein, based nanomaterials. The significant advancements where proteins are unfolded and reassembled into novel nanostructures with enhanced capabilities, such as enhanced encapsulation, versatile drug delivery loading,
and sustained release profile. The denatured state enables efficient encapsulation of both hydrophilic and hydrophobic molecules, because of when you denatured protein, hydrophobic and hydrophilic amino acids will be exposed, and they will capture both hydrophilic and hydrophobic molecules for more encapsulation. In this study, we encapsulated rhodamine as hydrophilic molecules and ANS for hydrophobic molecules. We choose these two molecules as model, and for confirmation, we can use and also drug hydrophilic and hydrophobic drugs too. But we, in scope of this project, we used only model molecule. This enhanced encapsulation, for example, denaturation model and controlled release profile show great promise for improving therapeutic efficacy and reducing systemic toxicity in cancer therapy. In our other work, positively charged F-nucleosine-based nanoparticles were synthesized and interacted with cellular exosomes to obtain coronal structures. And then cell studies were carried out. The obtained nanoparticles were examined with characterization method explained here. Some part of this studies performed in Mainz medicinal faculty in Germany. As you can see here, zeta potential of plain F-nucleosine, you can see here about plus 19 millivolts, and after treatment with exosomes, and as you know, exosome is minus degree, zeta potential of these nanoparticles decreased to approximately minus 7 millivolts. This confirmed that our dipeptide-based nanomaterials interacted with exosomes. And here you can see some transmission electron micrographs of these nanoparticles before and after interaction with exosome. There is not any particle size disruption and they stabilize their particle size. And here you can see the cell viability test performed with different cells. In this study, silicon-based nanoparticles were also used as controls and were also treated with exosomes. Considering the cell viability test of both types of nanoparticles, the bio-based nanoparticles appear to be non-toxic. Here you can see the silicon-based ones and F-nucleosine dipeptide-based ones. More better, it seems more better. In another studies, we synthesized diphenylalanine amide-based nanomaterials in different experimental conditions and obtained around 88 nanometer sized particles. Although these nanoparticles are normally negatively charged, we decorated them with calcium ions, and you can see that after calcium ions, the zeta potentials was changed.